So I will speak of plant morphogenesis. But before this, I will just introduce what we do in Fribourg in mathematics. So we work in applied probability on chemical reaction networks, statistics, multivariate analysis, and geometry. And mathematical biology, we focus on mathematical models on various topics like ecology, gene regulatory networks, plant morphogenesis. And in systems biology, we collaborate with two directions. One is on complex webs of bacteria, collaboration with Jan van der Meer. We have a synergia together. And we perform, not myself, but we have data from experiments and we try to model this. And what I will speak today, pattern formation in plants. I collaborate with Didier Reinhardt from the plant science department in Fribourg. We are actually performing experiments on plants and try to model some aspects of plant morphogenesis. So phyllotaxis is an observation of regular patterns in plants. Like you see three pictures, one cactus, one pine cone, and one sunflower. So we see that there are spirals and nice geometrical structure. We can find also here Fibonacci numbers. Here we see parastiches. Parastiches are the apparent spirals in sunflowers or in pine cones. And it turns out that there are always Fibonacci numbers. Here, clockwise and counterclockwise, we have two consecutive Fibonacci numbers. Okay. If we consider, for example, a cactus here, and we measure the angle between two consecutive leaves, it's more or less the same angle, the golden angle, modulo some stochastic perturbation. So what we see is that there are quite strong invariants in plants, geometrical invariants. And for example, why did evolution lead to, for example, spirals in sunflowers is to put seeds in the most compact way in the plant head, because to favor at most as possible reproduction. So, okay, so there are some reasons why nature found this structure. But so phyllotaxis was already present a long time ago in the antiquity. Here we see some capital with acanthus leaves, okay, and this scientist or philosopher used the phyllotaxis to classify plants, okay, 2000 years ago. And one typical kind of phyllotaxis is a spiral phyllotaxis where leaves turn around an axis. And it took a long time to find this structure. It was not so evident. So I just make a small history, a brief history of phyllotaxis. Leonard Vinci observed that there were some spiral. So we can find this text in one of these manuscripts. And Kepler too was the first to see Fibonacci numbers in phyllotaxis. So he saw that when we see a tree, then the structure of the leaves and so on show Fibonacci numbers. So next, the Swiss naturalist Charles Bonnet put in a strong basis spiral phyllotaxis. He explained that there was a spiral and so on. And later, so two botanists, German, make precise a relation with Fibonacci numbers and the golden angle. So, later, if you want, the first mathematical models, because our consortium is devoted to do theoretical biology or mathematical model in biology. And here we see what kind of mathematical model can we first establish to understand this structure. So a basic idea is that there is on the left here, there is a underlying generating spiral, and we put seeds, like to explain the structure of the sunflower in the sunflower. We put consecutively seeds along a spiral with the same at fixed angle. We see here an example, and this angle here is a golden angle. One can show that this leads for this special angle, we will have the most compact configuration. Okay, and but this is a first model, purely descriptive, and it has almost no biological content. Okay. But Hofmeister, not in 1868, was looking at the head of sunflowers, and at the head here of the sunflower is a so-called meristem, and in the center of the meristems you have stem cells which differentiate and migrate to the boundary here, and we see the old seeds already present, and the new incoming seed will be placed at the least crowded place. It's like I wrote perhaps because seed repel. There is perhaps the idea of Meister is that perhaps there is a repulsion force. Seeds repel. Later, so Turing was fascinated by phyllotaxis, and this is exactly in this context that he developed reaction diffusion equation to explain pattern formation. But it turns out that it is not really a good explanation, a good model for phyllotaxis. But the idea of Meister was very powerful. This led to a lot of interesting mathematical work. But again, these models are based on observation, okay? And here on a basic idea, repulsion. So you have a mathematician or physicist like Adler. Levitov proposed a model from statistical mechanics where he formalized the idea of repulsion and make a link with geometry and number theory. Later, o.d.e and Kuder, two physicists, proposed an experiment to validate Levitov model. And so he imagined an experiment where that leads to repulsion between droplets, magnetized ferrofluid droplets in a pool of silicon oil. And effectively, this repulsion force led to spirals, like we observed for example, in sunflower. Later, Kunz from Lausanne, I think, provided a rigorous mathematical study of the Levitov and o.d.e and Kuder experiment. A very nice paper published in Communication in Mathematical Physics. And later, not too far, Attela, Golay, and Houghton proposed a dynamical system approach based on the idea of repulsion. And again, they found a link with hyperbolic geometry and number theory. But in fact, these models are based only on the observation or the idea that seeds repel. But if we look at a real plant, so the meristem is located at the top of the plant. This is the basic place where patterning occurs. And we see on the second image here, primordia. And the growth in plants is due to a plant hormone called auxin. And these auxin molecules are the main actor of the plant patterning process. Here we see meristem, and in light green we see hotspots of light green molecules. These molecules are auxin molecules. So basically, when at some point or some set of cells, there is a high density of auxin molecules. This auxin molecule will attract enzymes that will modify locally the tissue, and perhaps with the pressure and so on, this will lead to a new primordium. So the occurrence of primordia are caused by auxin accumulation. But these auxin molecules move around in the plant, not perhaps randomly with a small amount of diffusion, but it turns out that they cross membranes between cells using pin proteins. So pin proteins are transporters. So to make a summary, auxin molecule can move around in the plant, and when there is a hotspot, like here, a new primordium will appear, perhaps a new seed or perhaps a new leaf, or okay. And so what we will see during this talk is some mathematical models to explain how auxin molecules are moving around in the plant. But as I said, to cross membrane, to move from one cell to the other cell, auxin molecule need a pin protein, and these pin proteins will stay in the cells, but they will be located at the interfaces of cells and help auxin to cross the membrane. So now I will focus on more mechanistic models, not only descriptive mathematical models. That is, models that take into account the biology. One of the first model is a model of Shipman and Newell, and they use the fact that the meristem is viscoelastic, and so they took von Karman equation from elasticity theory to explain the patterns using buckling theory. So it was a very interesting work. Using these very difficult von Karman equations, they were able to derive some elements of phyllotaxis like spirals, Fibonacci numbers, and so on. Later, Johnson and Smith proposed a model based on the auxin flow. So they wanted to explain the patterning process using auxin and auxin accumulation that lead to new primordia. I will discuss all this model later, okay? A concurrent model was proposed by a different group, Feugier, and they also modeled auxin transport within the plant, but using the flux. The first model of Johnson and Smith was based on concentration. This will become clear later. But basically, there are two different models, like which can explain some patterning aspects of plants using the auxin, okay? And later again, Newell, Shipman, Son took their first model based on von Karman equation, and this equation explained buckling, that is pattern formation in viscoelastic media. And when, I will not write the equation here, but this equation involves stress tensor, and this, in the new model of Newell and Shipman and Son, they included the auxin flow in the stress tensors. And so this is a mixture of mechanics, of viscoelasticity, and of biochemistry. And interestingly, they took the model of Johnson and took a continuous limit. And what they found is that the biochemical model of Johnson and Smith, which models auxin transport in plants, is similar to von Karman equation. It's very strange, but okay. So I have here a simple picture where we see two cells, and in green we have auxin molecules, in red pin proteins. So pin proteins are mainly located in the cell, in cells, but they can be located at the interface between cells. And when a pin protein is placed on the membrane, then it can transport an auxin molecule for a move from one cell to the other, okay? So we see here a real picture. We see in light green auxin molecule, hotspots of auxin molecules. And so we see that when we have a hotspot of auxin molecule, if we look around, there is no more auxin molecules. So the accumulation of auxin molecules creates a depleted zone of auxin molecule around, and this might be the origin of the repulsion force. Because as we will see, hotspots of auxin molecule attract in some way auxin molecules. And so if there is a void zone devoid of auxin molecules, the new auxin molecule will not come again, and so this will indirectly perhaps create a repulsion force. I am not sure, but this is just an idea. So here we see, so the first step, pattern formation in the meristem, as I explained. Here we see tomato plants and the golden angle. So first emergence of pattern in the meristem. And in a second step, the plant must evacuate auxin because it can become toxic. And so the plant is creating veins, mid veins, to evacuate the auxin. And this the process of developing new veins is also created by auxin. Okay, I will explain. This is basically the self-organization of the vascular systems in plants. I will use the second model of Fugil. So for the first phase, I will describe the model of Smith and Johnson based on concentration, and for the formation of the vascular system, the second model. So the model. So we see cells I, J, K, okay? We see pin proteins at the interfaces. So AI will denote the auxin concentration in cell I. PI is the concentration of pin protein within cell I, and PIJ is a pin concentration on the interface separating cell I from cell J. Okay, so we have basically three family of function of time. And so I will now describe the equation of Johnson. So the increase, the derivative of AI, which is the increase of auxin concentration in cell I, is due to mu. Mu, it is a production rate, but basically in the meristem it is zero because the cells are not producing auxin, they are coming from leaves. So, okay, but here basically we can introduce mu production rate and nu degradation rate, but we can suppose that both are zero here. We have a diffusion term. This is the third term in this derivative. This is just modeling a random walk on the graph of cells. So the set of cells form a graph. And this is a small term because it is known in biology that diffusion is very, very weak in the patterning process. But we can include a diffusion term. So the sum here, the sum of the K neighbor of I, this symbol means that K is a neighboring cell of I. So AK, this means that the auxin molecules present in cell K can move to cell I, okay? And the negative term means that auxin molecules which were present in cell I can move to other neighboring cells. And the main term, most interesting term for the patterning process here is the last term. And we see again a sum over all neighboring cells, K. For example, from cell K to cell A. So auxin molecule which were present in cell K, the AK here, this is a concentration, can move to cell I at a rate pKa, which is a concentration of pin proteins present at the interface Ki. Okay, and again, auxin molecule which were present in cell I can move to cell K. The second differential equation, the increase of the concentration of pin proteins located at the interface Ij is due to the cells, the concentration of pin proteins which were inside in the cell I, but moved to the interface Ij. And you see the rate at which pin proteins move to this interface is proportional to some function f of Aj. This means that pin proteins will move at the interface separating cell I to J with at a rate which is proportional to the concentration of auxin molecule in cell J. So the more F is increasing, here we take a linear function, okay? So the more you have auxin molecule in cell J, the more pin proteins will be located at the interface Ij. So in some sense, hotspots of auxin molecule in J will attract pin proteins. And this is a positive feedback. So if you have many pin proteins at the Ij interface, then molecules which were present, auxin molecules which were present in cell I will be transported to cell J. So we have a positive feedback. This looks complicated, but in fact, Johnson simplified the model by assuming that the displacement of pin proteins is very fast. So they took what it is called adiabatic limits. They suppose that the process is very quick. It reaches quickly an equilibrium. And if we do this, we get an equivalent model here. The increase of auxin molecule is due to diffusion and the transport as before, okay? But now we have this form for the transport PIK, the rate at which now, if we don't see here pin proteins, it's only a differential equation involving auxin. So we can interpret this term here, sum over the nearest neighbor K of I, AK, K, pKa, as the rate at which molecules move from cell K to I, the first term here. And we see this term is proportional to the auxin molecule in cell K. So again, auxin molecules present in cell I move to cell K having high auxin concentration. So hotspots of auxin molecule attract auxin molecules, okay? So when we do simulation, we see that such models can produce patterns. So now the formation of veins. What is the difference? If I see the first picture A here, explain what I did before, we see that pin proteins will be put in the membrane as function of the auxin concentration in cell J. In the second model of Feugier, because we cannot explain how, if you want, pin protein can know the concentration of auxin molecule in cell J. It is a completely unknown, no explanation. This is only a plausible scenario, okay? But here it is perhaps more realistic. The insertion of pin proteins at the interface will be the consequence of the flux between cell A and cell J. So they can perhaps better estimate the flux at the interface than to know the concentration of auxin in the other cells, okay? Perhaps they can measure, they have some way to measure this flux. So mathematically, the flux from A to J is defined as this term, which is intuitively clear. For example, the second term is just the amount of outgoing auxin molecules and the difference between the outgoing molecules that move from I to J and the reverse of the incoming auxin molecule moving from J to I. Okay, this is a basic flux, so like in physics, if you want. And if we look at the second model, which explains usually the vascular system, we have the first equation is the same, basically. But for the second equation, the rate at which the increase of the concentration of pin proteins at the IJ interface is due to some nonlinear function, phi, nonlinear response function, which is 0 when the argument is negative. So this means that, for example, if excuse me, when the flux is negative from I to J, that is, there are more auxin molecules moving from J to I than the converse, then the contribution here is 0. But if so, there will be no pin insertion in the IJ interface. But if this flux is negative, then the minus this flux, which is a flux from J to I, is positive. So the flux will be strongly directed. Okay, the pin will follow the flux in some sense. Okay, and if we consider a simulation, we can see that such models are able to explain the pattern formation process associated to the vascular system. So, why people do this or create these kind of models in plant science? Because it's very difficult to make experiments, and they need some plausible scenarios to understand the patterning process. And mathematical models offer a possibility to test hypotheses. At present time, nobody knows which of these two models is the best one. What I think is that it depends, and what many people think is that it depends on where the auxin are located in the plant and the nature of the tissue. Moreover, there exists 7 varieties of pins, and perhaps some pins are expressed in some part of the plant, and this will choose between the way of moving in some sense, but it is completely open, I think. Now I just provide an idea of the mathematical models. For the first model, I will not have to treat the time to treat the second model. So we come again to our mathematical model. So we have a different system of differential equation, and which are nonlinear, okay? Because you see the second term here. If we would have only the first term, this is a diffusion. So basically, what we will observe is a uniform concentration, okay, with diffusion. But because of the transport process, we have a patterning process coming to the game. So the system is conservative if I don't have production and degradation. So the total number of auxin molecules is constant. Okay, and when we have such a differential equation, okay, the first step is, what is very common to do and very useful, is to put the derivative to 0, because this is kind of steady state of equilibrium. So we put the derivative to 0, and we look therefore at the AK, the values of the various AK, such that these sums vanish. Okay, so we put the derivative to 0, and when we have, we can model. Here we have, for example, two two cells, for example, in 2 dimensions. When we have a dynamical system, a differential equation, we have a vector field, and we look at the critical point. That's where the vector field is 0. Okay, and so basically we have to solve an equation like this. 0 is the sum of the diffusion term plus the transport term. But we can re-express when we have no diffusion, but it is not unrealistic in biology, okay, to have no diffusion. It is known that the diffusion is very, very weak in the meristem, for example. So we can re-express the o.d.e, the ordinary differential equation, in a different way. So the derivative of A is some function of A, and the function is given by the above expression. But now we model to be close to Markov chain theory, we have introduced a new variable q, ki, okay, which depend on A. Okay, this function depend on A, so we can, using this, rewrite our basic equation. F of A is 0, as 0 is A times Q of A. And for people having followed course on Markov process, we can see that QA is a Laplace operator associated with a Markov chain, but this Laplace operator depends on A. So it's not very standard, okay? Because usually when we have such a steady state equation for a Laplacian Q, it does not depend on A. But we look for A, which an A in probability theory, it is known as the steady state distribution or the invariant measure of the Markov process. So if we call P the invariant measure of the Laplacian, we have to solve such an equation. A is PA. And where we were looking at this mathematically at this problem, we observed that researcher in probability theory working on vertex reinforced random walk, they arrived at the same kind of generator, more or less. Okay, so we were able to find the explicit form of PA and then solve the equation. But what is interesting is that basically mathematically, the self-organization of the auxin flow should be related to a reinforced random process, like a reinforced vertex reinforced random walk. Okay, so we observed what was done in this setting, and we found that the equation is solved for all configuration A's such that the sum here, the sum over all k close to I is constant. So I will not discuss too much on this equation, but just what is interesting for people doing either probability theory, discrete mathematics, this is just a simple equation involving the adjacency matrix of the graph. So basically, we have to solve. If I put, you see in this picture, we put in black component I such that the concentration of auxin molecule is 0, and in red where the concentration of auxin molecule is positive. Okay, so we have subgraphs where we suspect that auxin molecule, the auxin concentration is positive. So we have families of subgraphs, and on each we call these subgraphs gamma. Okay, and on each subgraph we can consider the adjacency matrix of the graph, and we can find the solution, the steady state solution, by solving an equation of this form. The adjacency matrix of gamma times A is a constant. It turns out that for example, for the square, for the cross, the solution is not unique because the determinant of the adjacency matrix is 0. This means that for a given configuration, we have a continuum of equilibria. It's not a single positive equilibria, but a continuum, okay? And now stability. When in the dynamical system theory, if we have an attractor, we can, a critical point, on the right side, we see an attractor, all orbits, all the flow is converging to this, the center here, and the center here is a solution of the equation FA is 0. And on the left side, we have also again a critical point, but it is repulsive. The orbit will not converge to this point. And to decide if a critical point is an attractor or not, we have to consider the Jacobian matrix of the system, so the derivative. And we've computed this Jacobian and found it is an expression involving the squares of the adjacency matrix. So, and to check stability, we have to look at the spectrum of the Jacobian, so at the eigenvalues of this matrix, and check that the real parts of the eigenvalues are negative. Okay. So we proved the following theorem. If I come back here, we see that positive concentration are in red, zero concentration in black. So having a graph like this, we can solve the equation to find gamma, the adjacency matrix times A is constant, and check if this equilibrium is stable. So we prove that if a building blocks, I call the building blocks these subgraphs, a component, as diameter greater than 2, it is unstable. So we perform some spectral analysis on the previous matrix, and we have then found that the two motifs on the right here are unstable, and the only stable ones are these 6 patterns here. So we can imagine that equilibria of the model are composed of such building blocks which are isolated in a background of auxin depleted cells. Auxin depleted cells is. Put a picture here. We have our stable building blocks, also a square, a simple segment. These are stable. They are a good candidate to be stable. But in order to be stable, this kind of limiting configuration as stable, we must have more. They must be far away. Far means that the distance must be at least 2. The neighborhood of a configuration must not cross the neighborhood of different subgraphs, if you want. You see here the distance between these 2 blocks of 3 cells and 2 cells is 2. So the difficulty with when studying these dynamical systems is that there is no single equilibrium, but a continuum of equilibrium. So we proved convergence. We proved that the system has a Lyapunov function, and that the orbits of the auxin flow converge towards the continuum manifold of equilibria, but we were unable to go further. It seems to be a difficult mathematical problem. And so when we see a true meristem with a confocal microscope, we can see that we have hotspots and we see some patterns, and we can imagine that perhaps the concentration-based model which produce typical patterns like this, these 6 building blocks here, which are stable, might perhaps explain this kind of patterns we can observe in real plants. So now I will finish here. I check the time. Okay. So what can we say concerning this model? First, I think for myself, I think that the model of Newell, Shipman, which mix together mechanics, von Karman equation, and the auxin flow is a very good way of explaining what is observed in plants. And that the biochemical process here based on auxin will perhaps furnish pre-patterns. That is, local hotspots of auxin molecules, and then to explain the real patterning process, one should use viscoelasticity in conjunction with biochemistry. Okay, so I will stop here because I will not have to speak of the second model. Okay, so I'm finished.